What would happen if you took blood from ME-CFS patients and actually exposed that blood to human tissue in a laboratory setting? Would that tissue remain the same? Or might it actually become dysfunctional in some way, thereby proving the biomedical nature of ME-CFS, but also giving us insights into its pathophysiology? In today's video, we're going to be talking about a groundbreaking recent study which has looked at just this question, and the results were pretty astounding. My name is Patrick Usher. I'm an ME-CFS patient and author of the book Understanding ME-CFS and Strategies for Healing. And in today's video, we are talking about a study out of Germany called ME-CFS and PASC, that's post-acute sequelae of COVID, patient-derived immunoglobulin complexes disrupt mitochondrial function and alter inflammatory marker secretion. Let me tell you that this paper is so complex that you could easily develop cognitive-induced post-exertional malaise. So what we're going to do in today's video is just talk about it in a very basic way. How was the study set up? What were its aims? What were 3 key takeaways from the study? And what would be my overall reflection on what it tells us. So first of all, what did this study do? Well, it actually took the blood from ME-CFS patients and it purified it down to just IgG, that is to say immunoglobulin class G, which is essentially a series of antibodies or autoantibodies which are created from the plasma and B cells. And it focused specifically on that class of IgG because it has been previously found to correlate with symptom severity in ME-CFS. So the more autoantibodies a patient has belonging to this particular class of immunoglobulins, the more sick they tend to be, the more symptoms of ME-CFS they tend to have. So this study, rather than looking at, what happens in response to ME-CFS blood in general, was very specifically focusing on the response that would occur specifically from these kinds of autoantibodies that have been implicated in ME-CFS for quite some time now. Okay. So it took the blood from some ME-CFS patients and it exposed it to human tissue in a laboratory setting. More specifically, that human tissue was actually taken from umbilical cords, and this actual use of umbilical cord tissue is actually something that is often done in scientific experiments. And in this case, they were exposing it to endothelial cells, that is to say blood vessel cells, from the umbilical cord. Now, you might ask, why are they doing this? Why not do the same study in humans? Just take a healthy human and give them the blood of ME-CFS patients and see what happens. Well, the problem, of course, with that is that you would just most likely make that person very ill. In fact, you could probably induce ME-CFS in someone just from doing that. We know, for example, that in mice studies, when blood from ME-CFS or long COVID patients is put into them, the mice actually develop ME-CFS overnight. And so the great risk is that you would essentially cause the same thing to happen in a human being, and that of course would go against every ethical standard for doing scientific research. And so we are instead left with these kinds of in vitro studies, that is to say studies done in a laboratory. And while they have limitations in that they can't tell you about the full complexity of, the interactions that would occur with the same experiment in a human organism, they can show very clear mechanistic outcomes that can give precise insights into the nature of the pathophysiology. And this is really interesting in relation to autoantibodies because even though, as I say, we know that they correlate with symptom severity, we know from the same research group that treatments that can remove autoantibodies like immunoabsorption can lead to symptom improvement very quickly. And we also know, from a textbook perspective, what the effect of these autoantibodies might be. And generally speaking, they cause reduced blood flow throughout the ME-CFS body, and they do interfere with cellular function. But what we don't really know is, at this very kind of clear mechanistic level, what happens when these autoantibodies actually, react with human cells. So that's basically the backdrop. They took the blood from the ME-CFS patients, they added that blood to the human tissue, and then at a cellular level, at a mitochondrial level, they observed what actually happened next. And the results really go a long way towards further elucidating the nature of ME-CFS and showing just how serious a disease it is. This video is sponsored by Turny, a very exciting new artificial intelligence platform which has been created with ME-CFS and long COVID patients in mind. Now, Turny has done something very different. It has actually indexed and incorporated pretty much everything to do with ME-CFS and long COVID from the last 10 years. That's all the research papers, all the conference talks, YouTube videos, podcast discussions, even discussions amongst patients in open forums. Everything has been incorporated into the platform. So when you ask Turny a question, it is drawing upon a lot more sources than, for example, ChatGPT, which tends to give useful but often quite vanilla answers. So if you want a platform that's been designed much more for your situation as an ME-CFS or long COVID patient, please check out the links down below. Turny is only $2 a week, but it's also free for the first 3 weeks, and you can get an extra 10% off if you use the code Patrick10. Okay, so these are my 3 main takeaways from this study. And so the first is that this blood from the ME-CFS patients actually led to widespread degradation of mitochondria and destabilisation of mitochondria. Now, to be clear, what was not happening was mitochondrial fission, that is to say, the actual breaking apart of mitochondria or cutting apart of mitochondria, which is more serious. But what was happening was a range of mitochondrial proteins, their output was reduced, and this suggested that the mitochondria were actually becoming, dysfunctional in some way, that their capacities were being degraded, and that they were essentially being destabilised. They were being put under significant stress because of the interaction with the autoantibodies. And this change occurred very quickly. This was happening within hours. And so there's this very direct link between the autoantibodies and how that interacts with the mitochondria and whacks them out straight away. The second key takeaway is then how the mitochondria were utilizing oxygen and creating energy. So in relation to oxygen consumption, it was actually observed that this lowered, again, very quickly within the mitochondria. And this then again ties in directly with what is so often seen in ME-CFS, which is reduced tissue oxygenation. So here we have yet another reason for that. But what was really interesting was that the mitochondria actually upregulated their attempts to create ATP, the key energy molecule in the body, adenosine triphosphate. They upregulated their creation of ATP from glycolysis, that is to say, using glucose to create energy. And this was really interesting because what it essentially suggested, according to the authors, was a kind of response to stress. In other words, once the mitochondrial function had been kind of degraded and destabilized, the mitochondria responded to that by recognizing we're in a bad situation. Because we're in a bad situation, we're going to need to try and actually create more energy because things are going wrong here. So they actually tried to make even more use of glucose and really squeeze the life out of glucose, as it were. And while this response is adaptive in the short term, if it's happening day after day and week after week and month after month, it's not something that the body can very easily sustain. And so what the authors of this paper noted is that they think that this phenomenon would ultimately lead to increased oxidative stress, increased production of reactive oxygen species, and in and of itself would actually lead to mitochondrial dysfunction. So from these first 2 takeaways, we have mitochondrial destabilisation, degradation, and this kind of stress response, which is in and of itself going to lead to further mitochondrial dysfunction. And clearly this kind of picture, this kind of pattern ties very closely into, for example, the work of Robert Navio, who has described this cell danger response as something that occurs in ME-CFS, that the cells kind of go into this shutdown or hibernation mode. And in fact, elsewhere in this paper, they also found a kind of hypometabolic and under-functioning metabolic state phenotype that was also occurring in response to the autoantibodies. And the final key takeaway, and on a slightly different note from this paper, was that it was the blood from female patients that caused a much more pronounced negative response. It's not to say that the male blood did not cause a negative response. It did, but it was much more pronounced in the female patients. But what this clearly does speak to is the long-established fact that more women tend to have ME-CFS than men. And so there may be something about the way the immune system, female ME-CFS patients, is malfunctioning that creates an even more negative effect with these autoantibodies that actually then can kind of kickstart or continue the disease mechanisms. So that is something that could go some way towards explaining this discrepancy between the gender balance in relation to ME-CFS. So those are, I think, the 3 key takeaways from this study. It does leave open other questions. It kind of noted in the study that the severity of the patient in question didn't seem to matter in terms of the mitochondrial response observed, but this was a very small study. It's a pilot study. It's proof of concept. And I think really what would be interesting would be to do a much larger study and then kind of tease out, if you, let's say you have 30 severe ME-CFS patients, 30 moderate, 30 mild, does the blood from these different groups more clearly affect the mitochondria and cellular function? I think that would be a very interesting study to do. But regardless of those kinds of open questions, this study is absolutely fascinating. And what it does is it clearly shows the biomedical nature of the disease. But it also tells us that the role that autoantibodies are playing is even more than we might have thought before. Now, not every ME-CFS patient has these autoantibodies. I do myself. I tested for them a few years ago with the Cell Trend lab when I was in Germany. But, I think for the patients who do have the autoantibodies, they really do create a very tricky situation, because I do think that they create an ongoing burden in the body. Whether you're having a good day or a bad day, things aren't right because the autoantibodies have burrowed into the tissues and they are causing, ongoing problems. But whereas before the overall picture was they worsen nervous system function, blood perfusion, and yes, they contribute to the cellular, what this study shows is a very direct, very immediate impact on mitochondrial and cellular function, which is actually very deep. And that, I think, only further goes to show that for patients with autoantibodies, these do represent a significant barrier to healing. Not complete barrier, I'm sure, but a more significant barrier. And I think another takeaway is, I think these kinds of studies are great. I think in vitro studies, despite their limitations, allow researchers to look at things that would not be possible to do in live human beings, and can really elucidate the mechanisms of the illness in very clear ways. And there's another study that I want to talk about on this channel soon, which did something similar, which actually took ME-CFS blood and exposed it to muscle tissue that was grown in a laboratory. And the results in that case too were absolutely fascinating. So I think this is definitely part of the way to go forward, and something that can just only further clarify the nature, and indeed devastating nature, of ME-CFS. So that's it for this video. Please leave your thoughts and comments down below. Please consider checking out my growing Patreon research and treatment support community. Again, links for that down below as well. But otherwise, I will see you in the next video when I think I'll be talking about a similar theme.